This video is going to be all about sensory processing and how autistics learn the fast rates of information coming in. We'll talk a little bit about sensory processing, but we'll talk a lot about mini-columns. We've mentioned mini-columns in the visual thinking episodes. So this is just drawing these things out because I know the episodes are very dense. And I want people to follow along though. I want it to be easy to understand because I want the autistic phenotype to be understood. Because these are real benefits, what the biology of autism gives us, if understood, and if put into environments and situations where they can utilize the autistic phenotype. So this is all about mini-columns. Over the recent few episodes, we've covered a lot of things regarding the autistic phenotype and the ability to learn and sensory processing, the fast sensory processing. I know it's pretty dense and so forth. We talk a lot about excitation and inhibition. The last 3 episodes were about excitation and inhibition more specifically. Episodes 4 and 5 way back were about excitation and inhibition as well. And throughout many different episodes, we've kind of woven in the excitation and inhibition kind of phenomena with autism. But since we've detailed it more and more, and we've talked about sensory processing, those high rates of information more and more, there's some things here that maybe can help us understand what is happening with the autistic phenotype. Okay, so there's some recent episodes that are kind of regions of interest for us. That's visual thinking part 1, which is mostly the neurobiology of visual thinking, and visual thinking part 2, which is how autistics learn. We talk about the machine learning, similar to AI, and how we build up different categories for different catalogs. We have massive amounts of databases to pull from. And in large part, that's because of what we're going to kind of get into today with the intense detail. And a large part about learning is paying attention to the information, kind of time on task. With the autistic phenotype and these high rates of information coming in, we'll talk about the gamma waves briefly before we're really going to talk about mini-columns, okay? That was mentioned in, I think, visual thinking part 2 episode, but we'll really draw them out and understand them a little bit better. But one thing with the autistic learning style is very structured, and that's huge for learning, right? That's when we're learning something, we have to have kind of an order to go about doing this. Otherwise, it's friction, a lot of friction inside, and it's very unstructured. The autistic phenotype learns in a very specific and accelerated way. And that's because the sensory information coming in allows us to not scan the information so much, but pay specific attention on the detail, on a specific detail. You'll hear about Temple Grandin talking about churches, and she'll talk about the different types of blocks or doors or something specific. And then from there, just with those databases, just massive amounts of imagery comes across, okay? This is another part about accelerated learning as well, because there's an added layer of visualizing. And this is massive because we are not only taking in the sensory information from the outside, but we're pulling from many different types of sources, if you will. And this is huge because this is increasing time on task and attention to detail. If you're thinking about learning, you have to learn the detail. It's not that complicated. So the biology that gives us autism really allows us to be here. And this is huge. So some of the episodes recently about excitation and inhibition that are kind of a region of interest is Sonic Hedgehog, inhibitory neurons, and autism. Okay, and let's see, thalamic reticular nucleus, sensory gating, and autism, and the excitation episode, go signals, and autism. These are the last 3 episodes that we've done. This is inhibition here, because inhibition is more of a region of interest for us. And then excitation here, which are mostly the pyramidal neurons, which we'll map these out in the mini-columns here shortly. So a way of thinking about excitation inhibition is our default is go. The whole reason we have a central nervous system is to move the living organism. So our default is go. We can't just always go, though. We need some sort of pull. And this is what inhibition cells are doing. It's kind of pulling back into an optimal range. If you think about excitation and inhibition, it's kind of like on a seesaw. Okay? Excitation here, inhibition here. You want a healthy balance. It's context dependent, depending on the activity. But the default for the autistic phenotype is here, high excitation. And in large part, from what we've covered, is we have low inhibition, especially the parv-albion interneuron. This is the most dense GABA cell that we have in the human body. Parv-albion interneuron. Remember, these are very fast acting. We have a whole episode on this maybe 6 or 7 weeks ago about parv-albion interneuron and the autistic phenotype, because autistics lack these, and especially in key brain regions like the insula, the medial prefrontal cortex is lacking. Kind of all throughout, though. So this is the very fast-acting GABA. This works on GABA A. Remember the different, here we talked about the different GABA receptors. We only have 3 GABA receptors. A and C are kind of fast, but GABA A is more dense. So we have more GABA A. These are ionotropic, which means they activate within 2 to 10 milliseconds compared to GABA B, which is metabotropic. Anywhere from 10 to 50 milliseconds. So there's a big difference there with the different types of inhibition. But this is our most dense one, parv-albion interneuron. So the autistic phenotype, there's not enough pullback from that go system. So there's always massive amounts of go. And that is a big benefit for learning, though. However, the outside world doesn't really go like we go. So there's a big contrast, okay? It's quite the conundrum with the autistic phenotype and, let's say, the classroom or just society at large. Remember the examples of bee movie, how the camera zooms out and you can see the hive and there's thousands upon thousands of bees swarming around at high rates, very efficient like. So we've also covered, and this is relevant, the waves, okay? The input coming in with the sensory processing. So different waves. So we have high gamma, and this is 80 plus hertz. Low gamma, so 30 to 80 hertz. Yeah, it's showing up. Beta, so let's say 12 to 30 hertz. Alpha, 8 to 12 hertz. Theta is 4 to 8 hertz. And then delta, which you're probably sleeping or some sort of psychedelic maybe in that state. But we also know from people like Earl Miller at MIT that we kind of have theta ranges going on in the background to help us task switch and modulate our attention. But not to get sidetracked here, the autistic phenotype, when we're processing sensory information, we live mostly here. That's because the rates of information are coming in at mass scales compared to kind of the normal typicals, if you will, which live mostly here with a little spike into low gamma. So with the sensory processing, for instance, vision, okay? So you have the retina to lateral geniculate nucleus, and this is a little relay station on the thalamus. All vision will hit here. And from here, it kind of just branches off. So each stop is just recruiting more and more brain regions, essentially, is a good way of looking at this. So from here, we can go to the superior colliculus. We've covered that in the autism and eye movements episode. This is a very cool region. It's a very fascinating, powerful region. We'll talk about the inferior colliculus soon with auditory. And then V1, the visual cortex, V1 through V4, making sense of what is being seen. And then things like the amygdala for the low road, and also the thalamus. It'll hit the thalamus. But it hits these regions, so let's say the eye, the retina here, sends it back to the thalamus, and on the back part is the lateral geniculate, right here. Lateral geniculate, and then the superior colliculus is down here. It's like a little noodle shape. In the midbrain, the mesencephalon is here. Okay, so we kind of have the pons, the brainstem. It's at the top of the brainstem. And this is the thalamus, kind of in the middle of the brain. So it will also go to the superior colliculus, will recruit back up to the frontal eye fields, which are up here. So if we had the brain here, cerebellum. Sorry, Dr. Reza Shadner, that's a bad drawing of the cerebellum. All right, and then if we just outline the brain here, the temporal lobe, we're coming in here like this, so forth. And then back here's the occipital lobe, right? So V1 through V4, it's kind of hitting here really fast. And then up through here is the parietal. And remember the visual thinking episode. There's a little nucleus here, about right here, called the precuneus. And in visual thinking, this is lit up. In the default mode network, this is lit up, especially in the autistic phenotype. And here are the frontal eye fields. It's right here with the sensory motor, but the anterior part of the sensory motor. So the superior colliculus here has to reach all the way up to here, and that's a distal connection, and that's what I'm talking about. This is where we lose eye gaze. Eye contact and eye gaze for the autistic phenotype is not that difficult to understand, because remember the analogy. If we have a destination and let's say there's 5 stops. From point A to point B, you make 5 stops. And essentially this is what this is. Stop 1, stop 2, stop 3, stop 4, and so forth. The autistic phenotype hits these early spots really fast, much faster than normal processing, because that's the high and low gamma in comparison to the beta and alpha. So the autistic phenotype is just getting blasted with these early sensory processes. And then by the time it has to recruit these distal areas, which is more cortical, it's challenging because there's a delay now because of this myelination here that connects these. So right in here is the medial prefrontal cortex. We talk a lot about that. This is huge. This is adaptive responses. When you're navigating the world and you're making good decisions and healthy decisions, and you can fit in with the ever-changing flow of society, your medial prefrontal cortex is leading the way. and also the dorsal lateral, which is more up here, and the orbital prefrontal, which is right here, just right here. So orbital, kind of medial in the middle, and then this outer surface is the dorsal lateral. This leads the way, and it will instruct the subcortical areas on what to do, how to respond, and so forth. Okay, that's a long wind about the sensory processing. But the thing to know here is these early regions of the sensory inputs coming in are just so fast for the autistic phenotype in comparison to so-called the normal or the typicals. Okay, that's a big thing. And that's one of two things that is about this accelerated learning type of thing. And the second thing is going to be the mini layers or the Minicolumns. So remember, the myelination is a problem with the autistic phenotype. Huge problem. Things like speech and language with the arcuate fasciculus. We also have another fasciculus called superior longitudinal fasciculus. So now research is kind of thinking that the arcuate is just a type of superior longitudinal fasciculus. But regardless, fasciculi are nerve fibers that connect distal regions. Okay. And with autism, you can just think about myelination, right? We've talked about the comparison of driving on an interstate versus a gravel road. Good myelination is like an interstate where you can have optimal rates of speed. The gravel road is from point A to point B is going to be different. And that's a big part of this. So the Minicolumns. Let's talk about the Minicolumns. And we have many Minicolumns. Okay. These are layers in our cortex. And our cortex is layered, kind of like crepes. So they just kind of stack onto each other like this. This is exactly what they kind of are. Layer 1, 2, 3, 4, 5, and 6, which is I have laid out here because they all have different roles. So we can draw this out. They will look like this. This is just a representation of it. There's different areas of many columns have different amount of cells. So like the cortex has, so I don't know, 100 neurons per just a little slice of area, like within microns. So we'll draw it like this as well. Let's see. We'll go like this because 2 and 3 is going to be crucial. Layer 4 is going to be its own like standalone thing. And layer 2 and 3 are full of pyramidal neurons. They look like pyramids. Remember? They look like pyramids. This is our go signal. These are excitation. And then also in here, you have parvalbuum. So they're kind of stopping unwanted signals too. There's this parvalbuum here. Layer 4 is huge because this is the input area. These are stellate neurons. They look like stars. We also have them in the pancreas and liver, but they have different functions. So these kind of star-like things right here. And then layer 5 and 6, as we're working down, it kind of looks like layers 2 and 3. Okay, and there's also some parvalbuum here. There's also other types of GABA. Somatostatin is a little bit. Remember, those are the second most dense. They're kind of here. Carotinin can be here. But these Minicolumns are found all throughout, like each cortex, like the visual cortex, the auditory cortex, the somatostatin cortex. all of these things. The cerebral cortex has their own, and they all have different roles. So there are some inhibitory neurons down here as well. And then these are columns. So they're like this. And you can just stack different columns together. So you can also have, these are just spatially represented like the same. Okay, then you have this starlet. And then down here, same thing. Some inhibitory, okay? So each one of these, this is one mini column. And as you stack some, if you just have a slice or splice, that's called macro columns. But we're just talking about one. But I also want to talk about the space in between, neuropil. This is where there's no cell bodies here. There's actually no cell bodies here. These are all axons and fibers and so forth, okay, for the connections. With the autistic phenotype, and this is big, this space here, let me clean this up a little bit. This space right here, if you ever look at drawings, diagrams, blueprints, or so forth, you might see some measurements that kind of look like this. This inside diameter, okay? In autism, this is smaller versus typicals. The density of these macro columns, because of the stacked and close together of the mini columns, is so dense. You can just find research articles and images just having a splice, and it's just full. The autistic phenotype is just loaded here with different cells. And remember, there's a decrease in inhibitory, and that's a big part of this as well. So there's massive amounts of pyramidal neurons here for the autistic phenotype. And this closeness together, it makes it hard to kind of shut off and remember the role of so excitation, the go, and then the inhibition, the thing that pulls us back down into healthy states, healthy ranges, keeps us out of GABA and so forth. This is huge in autism. The density here with the autistic phenotype, a lack of cell pruning. Remember the autism sensory map and serotonin episode. Serotonin's role in development is pruning the thalamus and these cortical areas of dendrites. And also in the autistic phenotype, the dorsal striatum has massive amounts of dendrites. Dr. Hannah Stevens at University of Iowa has studied that. She's a previous guest. I was going to say something, but I know what I was going to say. So you would think how the brain functions and how Mother Nature just allows us to evolve to be optimal. You would think these would be linear, either starting from 1 to 6 or 6 to 1. Okay, that's not the case. Inputs come in to layer 4. These are mostly inhibitory, depending on where you are. Definitely in the cerebellum, they're all inhibitory. All stellate neurons are inhibitory. In other areas, the different visual, auditory, somatostatin, somatosensory, I should say, and the cerebral cortex, they're a combination of excitation and inhibition. Now, you probably know that autistics have fewer inhibitions here. So it's hard to kind of dictate and determine where signals go. And that's a big part. So after inputs come into layer 4, inputs are sent up to layer 2 and 3. And then, so 1, 2, 3, essentially. This is the process. So why 2 and 3? Why not go here and down? Layers 2 and 3 is where you can say this is where our intelligence, our memory, our ability, our experiences live. So inputs come in from the thalamus. It will check layers 2 and 3 so that the specific living organism can draw from its database and go down to layers 5 and 6. Now you notice there's a 5A and 5B and a 6A and 6B. 5AB is mostly regular spiking. We've talked about tonic, phasic, and burst firing. This has a lot to do with the brain waves mentioned earlier. Layer 5A is regular spiking. 5B is intrinsically burst firing, okay? Where we need to really activate, we burst fire. So these action potentials will burst like this. Let's just draw this out right now. So this is tonic. This is just your baseline, okay? And this is also time. Time and baseline. So this is activation. This is like gamma, way up here at the top. So burst firing will go boom, 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 and then drop off, boom, boom, boom. Spike, drop off, boom, 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 like such. Now the delta is, if there's more increase than decrease, it's going to stay higher. Now as you relax, calm down, time passes and so forth, the burst firing can start to drop. You see, that's how our molecules work, essentially. Things like dopamine and so forth. Dopamination is good on this, from Dr. Anna Lemke. It's easy to read information. If I can read it, you can read it. I promise you. So this is what layer 5 is doing. Regular and burst firing. Layer 6 is kind of the output area, okay? Layer 6A is corticothalamus. This is huge for the thalamic reticular nucleus. Remember the thalamus is a shell over it, and it's all GABA. And a large part, it's part of albumin. So 6A is speaking back, corticothalamus. 6B is cortex to cortex. Cortex. So it's just speaking back and forth throughout the areas of the cortex. So the thalamus is huge. It's every sensation runs through there, and all of our models, how the living organism acts and lives, will run through the thalamus, because that's our sensory information. It's recruiting. Remember the basal ganglia. Maybe we'll do a drawing of basal ganglia too, and more detailed about the sensory processing. But just a side note, at Stanford, Carl Diceroth and a neurosurgeon named Jamie Henderson, this isn't autism related, but they found in humans that when people dissociate, or they're having seizures, or people that have bad trauma will dissociate, they found that the timing from these layers and information coming into layer 5 is off. Layer 5 and timing looks like to be the spot for when and why people disassociate. Remember, all of this is about timing, space-time. We covered space-time so much in this little rant that I'm doing, in these awful drawings, maybe. I hope you can follow along. But these are mini columns. I think I'll do a solo episode just about mini columns now that we've talked about the excitation inhibition in more detail. And we've covered a lot about the sensory processing, because the minicolumns are huge because of the amount of density and the inability to shut off. But there's huge benefits to that with learning. Much different than other people. So, minicolumns.